So our last speaker is Dr. Damon Lesmeister. He's the principal investigator of the Predator Prey and Bioacoustics Lab in Cornwallis, Oregon. He's a research wildlife biologist with the USDA Forest Service's Pacific Northwest Research Station and holds courtesy faculty appointments with Oregon State University's College of Forestry and the College of Agricultural Sciences. He's a co-advisor on graduate students with the OSU facility, and he studied avian and mammalian predator and their prey in various forestry ecosystems.
Happy Star Wars Day, everyone. And keeping with the theme here today, I'll randomly and out of context include some AI-generated spotted owl images as Star Wars characters. I'd like to thank the organizers for the invitation to present at this webinar series about our work to study and monitor northern spotted owl populations using bioacoustics. As I go through this talk, I will be presenting findings from many studies and won't be able to dig into the details. So at the bottom left, you'll see some QR codes on some slides that link to papers with more information. I'll also include my email on the last slide, so please reach out if you have any questions. Here is the hero of our story. The northern spotted owl is one of 3 subspecies of spotted owls. Their range covers the forested mountains of the Pacific Northwest from southern British Columbia in Canada, western Washington and Oregon, and northwest California in the United States. They are a highly territorial species with high site fidelity. They're non-migratory and have high mate fidelity, often mating for life. They are a quintessential case-selected species with high survival and low fecundity. Spotted owls are an old forest obligate, which has caused conservation of this species to come in direct conflict with human interest in harvesting old forest. A long history of logging these ancient forests in the Pacific Northwest with European settlement of these areas. And photos like these can give some sense of the size of these trees and what it may be like to be in these forests. But really, you can only fully appreciate this with close inspection and in person. We now have 5 decades of science, conservation, and conflict with spotted owls as the focus. My predecessor, Eric Forsman, shown here with a spotted owl, began studying spotted owls in the early 1970s and discovered much of the fundamental biology of spotted owls, especially this association with old forest, that populations were declining, and this was due primarily to large-scale logging activities. In the 1980s, there was significant conflict between the timber industry and conservationists in the Pacific Northwest. If you're interested in this history, please check out this multi-episode podcast by Oregon Public Broadcasting called Timber Wars. And you can see the QR code here. Due to habitat loss and declining populations, spotted owls were listed as threatened in 1990 under the U.S. Endangered Species Act. Then in 1994, the Northwest Forest Plan was signed. It guides over 25 million acres of forest management on federal lands. The plan transitioned federal forest management from being primarily timber harvest focused to ecosystem management that included many late successional reserves and also mandated the monitoring of spotted owl populations. In 1999, the monitoring plan for spotted owls was published. It laid a 2-phase plan for monitoring spotted owl populations. The first phase would use mark-recite methods to estimate demographic trends on these 8 study areas and then infer those results to the total population. The second phase would rely on habitat monitoring and range-wide occupancy rates to estimate population size and distribution. During phase 1, methods entailed callback surveys and mark reciting of individual spotted owls. Meta-analyses were conducted every 5 years, and each of these indicated that spotted owls continue to decline dramatically range-wide and to critical conditions in some study areas. Here you can see population change estimates indicating a negative trend on each study area. In 2020, the U.S. Fish and Wildlife Service determined that spotted owls weren't uplisting to endangered due to persistent population declines and unresolved threats. The primary threats to spotted owls are loss of old forests through timber harvesting and large wildfires. Also, invasive barred owls have fully invaded the spotted owl range and through competitive dynamics have contributed to declining spotted owl populations. Here's a figure showing occupancy rates of barred owls in yellow and spotted owl occupancy rates in purple. On every study area, barred owl occupancy has increased as spotted owl occupancy has declined. We now have mounting evidence that some spotted owl populations are in an extinction vortex, and this is a feedback loop of small and unstable population conditions that further lead to population declines and ultimately extinction. As I mentioned earlier, spotted owls are a territorial species with high site fidelity, so increasing adult dispersal rates, distances moved, and inbreeding are having a negative and destabilizing effect on populations. Population conditions highlight the need for adaptive monitoring that can change with declining populations and explore alternative methods that are less invasive than callback surveys and mark resighting. A couple questions that we had back in 2016 were, one, can we use bioacoustics to effectively detect spotted owls and barred owls? And two, can we use bioacoustics data to monitor trends in spotted owl populations? All of this also would have the potential for added benefit of monitoring many other species. There are many advantages of using passive bioacoustics for long-term monitoring. I've listed a few of those out here. We're really interested in bioacoustics because spotted owls as a territorial bird communicate extensively through vocalizations. Especially important in the context of population monitoring is the 4-note territorial call. Here is a male territorial call. We're also interested in pair status because it provides more information about the population. And so here's in a catalog communication. So you can see that we got pretty powerful information that we can learn about the species status at any particular site just through passive acoustics. While there are many advantages of bioacoustics, there are some significant challenges and that we needed to consider and overcome for the program to be successful. One is the startup cost, and I'm sure anyone on this webinar knows how expensive it can be to start a project that will cover large areas. For us, we were challenged because bioacoustics couldn't produce the same type of data that the demographic studies could. So no survival, no recruitment, no population growth, but rather the population metrics would be based on site colonization, extinction, and then change in landscape use rates. Also, big data is a challenge for any program for management, processing, and storage. We knew that listening to all the hours of data, even in relatively small scale, would be impractical. At the time, we had no auto ID models available, so we needed to partner with computer scientists to develop complex machine learning algorithms to facilitate auto ID and data processing workflows. Layla Dukoch, a former master's student in my lab, started in 2017 to tackle our first question about detectability of spotted owls and barred owls using bioacoustics. She found that we could achieve high detection probabilities of both spotted owls and barred owls, and also we had early evidence that we may be able to infer pair status because when paired, males were very vocally active. And as expected, detection probability decreased with distance from activity center. But the rate of decrease is important to know. And you can see this figure here with distance along the x-axis and detection probability along the y. And as you get further away from the activity center, we have a decline in the detection probability. Layla also observed calling patterns that were not known prior to this study. And as an additional benefit to this study was it helped us to inform and establish the most effective methods to survey a 5 square kilometer hexagon with high confidence that you could detect spotted owls if they're present. The next major question was, could we use bioacoustics to detect changes in spotted owl populations? So we approached this through simulations of 54 scenarios with 100 iterations each. We also were interested in to know what would be the most effective range-wide sampling design to detect a 4% annual population decline within 4 years. We measured both the accuracy and the precision of estimates to evaluate each scenario. Here are a few examples of sampling designs. The federal lands are the color polygons on the figures with different agencies represented by individual color. Note the size of the sampling area, 1,250 kilometers north to south, and the federal lands alone cover over 100,000 square kilometers. So regardless of the sampling strategy, we knew that annual surveys are going to be a major logistical challenge. The black dots that you see on these figures are the 5 square kilometer hexagons, and each of these have 4 sampling stations. We simulated different densities, and some options had a mix of sampling densities. For example, the 2 plus 20% option is 2% sampling on all federal lands, and then denser 20% sampling on the historical study areas where we have this long-term spotted owl data, which also has the added benefit of long-term research questions that require dense sampling. We found that the 2 plus 20% sampling design was the most cost-effective option. We were balancing robust sampling with high confidence in detecting population change, also with the ability to conduct research within a monitoring framework, and funding constraints. Based on these findings and other research, the Northwest Forest Plan Executive Committee made the final decision in December of 2020 to transition from Phase 1 to Phase 2 bioacoustics monitoring. Under the transition plan, we also have 2 years of overlap of demography and bioacoustics data. The red ellipses that you see here show where we have been conducting and expanding bioacoustic sampling. In some places, we've been doing this sampling since 2018. Demography ended in 2022, and this year we are in full bioacoustic sampling. And we estimate that this year we'll be sampling approximately 5,000 stations. We're going to be collecting about 2.5 million hours of sound, and we'll amass about 1 petabyte of wave file sound data. And this brings us back to an early challenge that we talked about in our work for developing machine learning tools to help process these data. Zach Ruff has worked on this project since coming on to help with Layla's master's thesis in 2017. He has held various positions and is currently an OSU faculty research assistant. Zach has successfully taken on the challenge to train several versions of a convolutional neural network that we call PNWCnet that automates data processing and detection of spotted owls and many other species. We've published a few papers on this, and another will be released soon. PNWCnet has allowed us to massively scale up our sampling and maintain rapid data processing with minimal human supervision. He also developed a R Shiny app for the PNWCnet that can be run on a standard PC. See the QR code here for the GitHub repository with everything you need to get started if you're interested in this R Shiny app. PNWCnet has gone through several versions, with each increasing in the number of species identified and improved performance for existing species. Zach is training version 5 and will include over 100 target classes. and which is a massive increase over version 1, which included only 6 species. Kara Apple, one of my PhD students, recently published this paper where we estimated detectability and occupancy rates of single and paired spotted owls using bioacoustics data. This is a multi-state occupancy analysis with the states being absent or used by a single owl or occupied by a pair. A goal for this analysis was to help interpret survey data and establish a framework for long-term monitoring analyses. I'll briefly go through the data processing workflow because this also applies to the next analysis that I'll talk about. We collect the data using ARUs, these autonomous recording units. We create spectrograms. We process the data with the PNW CNET, and the spotted owl detections were then run through a model to determine sex. Then we assign pair status at a site, and then we conduct the multi-state occupancy analysis. Notice here that the females, the spectrogram on the top, have higher frequency calls than males, and the males also have longer duration in their calls. Again, I'll go through the states of the multi-state models, given that they also apply to the next paper that I'll talk about. And we have the true state on the right, and the site can be either unused/absent. It could be used by a single owl or occupied by a pair. And then when we conduct these surveys, we may not have detected the owls even if they were there, and or we could have detected only one individual of a pair. So this imperfect detection in our observation is an issue that occupancy models have been developed to account for and estimate. Please note that the estimates that I present are weekly for a single sampling station. But what's most important is the comparison between these estimates. When we conduct these surveys, we're doing this for multiple stations within a hexagon, and we do the sampling for 6 weeks. If a site is used by a single owl, we have a 0.04 probability of detecting an owl during a 1-week period. So it's fairly low detection probability, but this probability increases when you include all 6 weeks of the sampling and include all 4 sampling stations within a hexagon. If the hexagon is occupied by a pair of spotted owls, we have a 0.18 probability that we'll detect at least one owl during a 1-week period at a station. And this, of course, increases considerably when we include all sites and all weeks of our sampling. The interesting finding here on pair occupancy is that we are far more likely to only detect the male. Splitting the 0.18 probability, 0.15 comes from the probability of only detecting the male, and only a 0.03 probability that we detect both the male and a female. And this has some really important implications for interpreting survey data when only a male is detected. Our findings suggest that in most cases, when a male is detected, it is actually paired, and you've just not detected the female. Females are much quieter when on the nest and stay much closer to the nest and activity center during the breeding season when we're conducting these surveys. When considering this study in the context of the other studies that we've conducted, is that now we have a really powerful toolbox to monitor spotted owl populations non-invasively throughout their range. We can now combine the power of bioacoustics data, artificial intelligence, forest inventory data, remote sensing, and multi-state occupancy models. And for this particular study, we found that high detection probability of residence, especially when paired, but we're most likely to detect only the male. Indeed, there was about a 72% chance that a site with only a male detected was actually occupied by a pair, but the female was just not detected. Barred owls in this study were also ubiquitous, and we also found that there was a negative effect on spotted owl occupancy by barred owls. Matt Weldy, another PhD student in my lab, published this paper recently where he integrated the bioacoustics data with historical territory occupancy survey data. The objectives were to resolve the spatial mismatch in these data sets, integrate the data sets to derive estimates for singles and pairs, and establish a framework for the transition from demography studies that use territory occupancy in the bioacoustics monitoring program. And we use the same data processing workflow as the Cara Apple paper. In this figure, the probability is along the x-axis. We have estimates for the probability of use, the probability of pair occupancy given use, and the probability of pair occupancy. The bioacoustic estimates are in blue, the territory survey estimates are in orange, and the integrated estimates are in purple. The bioacoustic estimates tended to have higher confidence intervals, but the territory surveys had higher pair given use estimates, which makes sense given the protocol to walk into nest sites after having detections at night to locate both the individuals of a pair. In this study, we were able to resolve the spatial mismatch challenge and demonstrated that this integrated multi-state occupancy approach is effective for harnessing the strength of both data sets. And we're also confident that this framework will aid our monitoring program in its transition. It is exciting that this monitoring program will remain relevant and cutting edge while maintaining linkage to historical data. One last study that I'd like to talk about is ongoing, but we do have some exciting preliminary results. In 2021 and 2022, we conducted what we call the Big Grid study, where we located pairs and found their activity centers or nest sites and set up a large 37 survey station grid with 1 kilometer spacing between these stations. The grids were centered on the nest tree or activity center, and we were seeking to understand the vocal activity patterns of pairs and do this non-invasively without the need to put GPS tracking devices and sound recorders on owls. We have data now for 8 sites. 4 of these are in 2021 and 4 in 2022 in a variety of locations. The map here in coastal Oregon mountains shows that the Big Grid sites, these are the black dots in the survey stations. You can see where these big grids were located in relationship to the randomly selected hexagons for population monitoring shown in green, which we also were conducting during this time. Some remarkable findings here. These are the 4 sites that we surveyed in 2021. The distance is along the x-axis, and the proportion of nights with detections is on the y-axis. The males are shown in blue and the females are in red. 94% of all calls were within 2 kilometers of the nest or activity center, and that further restricts to 98% of the female calls were within 1 kilometer. This has really important implications in that if a female is detected at a sampling station, it is likely quite close to a nest or activity center. We have more to come on this study when we're including the 2022 data, and we hope to have this published very soon. I have so many people to thank. It'd be impossible to name them all, and there's a few names here that people that have played critical roles in various aspects of what I presented today and our work toward transition this iconic monitoring program. And I'm really looking forward to hearing questions and having discussion in this webinar. Thank you.
Thank you so much, Damon. I'm glad as well that you've managed to get some more Star Wars references in there. I kind of really appreciated all the lightsabers and everything else. So thank you so much for that. There are questions coming in thick and fast as well. So hopefully we've got Damon on the line and we'll be able to ask some of those. Just
yeah, yeah.
Right. So one thing that I think people are having problems with, and either you can do this or we can help you do this, but someone said, please can you share the QR code links in the chat for those who can't access it? I think people have been really wanting to know lots and lots more. So if you need us to do that, we can happily do that. But if you want to do that, then please do. I think obviously there's so much interest in what you're doing. So you've got some controversial questions. The first question. I realize this is a wildlife acoustics webinar. Never starts good when that happens. But in my experience with the CSO, always involved recordings from Cornell. What are your thoughts on the different types of recording unit? Also, can we use Kaleidoscope with recordings from Cornell's ARUs? So, have you used the Cornell one?
We have used the Cornell Swift the Swift units, and they also are recording. It's a similar microphone quality, and yeah, so we have actually used that previously. But most of the units that we use are the SM4s and also the minis. And I'm not sure why my video is not working, but.
Mine was doing something really strange today by just connecting to my mobile phone rather than my computer.
Oh.
Thank you. You will indeed have epic data levels. What was the R app's name? Is it R Shiny? In other words, yeah. What is the R app's name and how new is it?
Well, the R Shiny apps, it's a GUI interface, and so a lot of people are using these, and you can use them for various purposes. And what Zach has done in the development of the R Shiny app specific to this project, we have the algorithm, which is the PNW CNET, this convolutional neural network, and you can install. It takes some time to get everything installed, but once it's installed, it's very user friendly. And yeah, it can be really highly effective. And we have very high performance for many of our species, especially for the owls. And we're into hundreds of thousands of training data that we've used for spotted owls. So the model performs really really well for spotted owls. And I'll say that in version 5, which it's in active training right now, will be somewhere around 80 species or so. But these, of course, are all in the Pacific Northwest of the U.S.
Yeah. Have you been able to determine whether barred and spotted owls are interbreeding, and if so, how much?
There are hybrids, and they do interbreed. I don't know that we have to my knowledge, we don't have a good handle on the rate of interbreeding, but it does happen.
It does happen. Excellent. Can you do this? Is a good one. I do like this. This is I get asked this with bats basically quite a lot. Can you distinguish different individual spotted owls from their vocalizations?
That's a great question. This is something that we are just starting to work on now. Matt Weldy he's one of the students that I highlighted there with one of his papers, and one chapter of his dissertation is going to tackle this specifically, this individual ID. And we do have some very early preliminary results that are, I'll just say, that are very exciting, and that there have been I think a lot of folks think we can't do this, but we certainly, if we are able to determine the number of individuals at a site, so it could be, you can imagine, we can distinguish male and female. It could be really powerful to know whether you have one or two males. Very unlikely you would have more than that. And it can also be really powerful if we know that we detect, individual A at this site or versus another site. So being able to detect the same animal in multiple sites can be really important information. And then if we are detecting the same individual in multiple years, so it will open up a ton of possibilities. And then you can sort of refine that, thinking about space. Of course, some animal that's many hundreds of kilometers away, if they may sound very similar, obviously it's not the same individual. So I think, yeah, using sort of multiple lines of evidence, we're confident, and we're confident that we're going to be able to determine, like number of individuals, and then and there are some other studies some early studies that have been successful at distinguishing individuals, and some individuals do have very distinctive calls. That certainly will help.
Excellent. I think you've answered the first part of this for the grid study, which ARU models we're using. But the second part of the question, why did you choose 1 kilometer? Sorry.
Oh, for the big grid study. That was the question. So there are one, we had some idea that we wanted to be able to go out about 3 kilometers. We thought that 2 kilometers is a pretty important distance, it seems, for paired owls, but we wanted to extend beyond that and sort of refine our estimates, and then be able to. We're not trying to locate the where the individual is calling from, but just about distance and sort of these patterns of calling behavior away from the nest. And so to minimize detecting a single call at multiple sampling stations, and we were not trying to use it, triangulation or anything like that to get the location of the individual necessarily, but how far away from the activity center would we be recording them?
So it's a very funny question, but I was thinking this as well. Hi, Damon. I get shivers and sweaty palms thinking about the humongous amounts of raw data you must have accumulated. How do you handle this? And not only store it, but to create an overview and structure for it. It must be so tempting to look at other species in the data. It can have many other uses, I'd think. So yeah, how on earth do you deal with these sources of data?
It is a challenge, and we've learned a lot over the last several years and made some pretty massive improvements, and there have been a lot of investments in having permanent positions in the program now, but we have a protocol, and this is something that we're actively working now to publish this protocol that will also include some of the data management, some considerations when we're dealing with massive data. And there are some unique aspects of it. And we also, some of the important considerations that we're thinking about is making these data a permanent repository, and that would be available not only today for many of these other biodiversity type questions. And that's where we see the program transitioning from being just primarily spotted owl focused, but really into a biodiversity monitoring program. So our goal, our long-term goal, is to be able to develop tools to extract vocalizations from all vocal wildlife, and so mammals, birds, everything.
Sorry, go on. Sorry.
Yeah. Oh yeah, and this would be accessible, potentially decades from now. There will be certainly questions that will come up that I don't think that we can have the imagination to think of today.
No, quite true. Actually, we've only got time for one more question, but it links on to that. Is your Shiny app able to be used for other bird species? And is it public available for other researchers? And can you train it for other species of interest? I suppose you've started answering that already.
Yeah, it is. Right now, what is in the GitHub repository right now, it is for the PNW CNET version 4. And this is 37 species that are included in that. So it will process not only spotted owls, but many other species. So like barred owls, marbled murrelets. Marbled murrelets is an important management species. We have some other state important species like sooty grouse, for example. So the Shiny app can be used for all of those species, and we have the performance metrics that are all included there also. So you have some idea of the confidence of the output from the neural network and how much effort you would need to put into validating that output. So that's part of our data processing workflow also, is this validation, especially before we use these data into any of our ecological modeling. We actually do have human observers that are validating all of that output.
Yeah, you need to know it's doing it right. That's the main thing.
Yeah.
Well, thank you, Damon, for a wonderful presentation. I loved all the Star Wars references, as you could probably gather.